Today, let me start the event. We have Professor Shubha Tole from the Department of Biological Sciences, who is going to present the science behind the Nobel Prize in Physiology and Medicine. Shubha, could you share your slide, please?
It is my great privilege to present the work behind the Nobel Prizes this year for Physiology and Medicine, awarded to David Julius and Ardem Patapoutian. The work that's awarded this year was for the mechanism underlying the perception of touch, pressure, pain, and heat sensitivity. Broadly, sensory perception is the key to our interaction with the world. In fact, sensory perception is how we know where we are in our universe and how we interact with them. And we know what the world is telling us. And this has certainly been the object of fascination for scientists and neuroscientists for centuries. And indeed, breakthroughs in understanding sensory perception have been awarded the highest of recognitions, beginning, for example, with the mechanism of auditory perception, von Besecki in 1961, then phototransduction, Wold in 1967, olfactory receptors, Buck and Axel in 2004, and then the Kavli Prize to Jim Hudspeth, Betty Place, and Pettit in 2018. Now, today, we're going to talk about different sensations than these, but broadly, for any sensory perception, the first question is, what is the receiver? When there is a signal out there, be it light or sound or a chemical or mechanical, what is the receptor? What is the receiver? And once there is a receptor identified, one has to ask, how does that receptor trigger an electrical signal? Because fact is, these receptors would be on the surfaces of neurons or on the very tips of the dendrites of the neurons. How do they trigger ion channel flow so that there is electrical activity in the neuron? Now, here, for example, is a cartoon of a receptor which receives this red signal. And once this receptor is identified, then one asks, well, how does it trigger flow of ions? And in this case, this receptor sets up a secondary messenger system whereby some other entity, that is the ion channel, then opens and the ions flow through across the cell membrane of the neuron. Now, this is the case actually for olfactory receptors and indeed phototransduction. The entity that receives the signal is different than the entity that ensures the flow of ions. There is another possibility, of course. The entity that receives the signal, which is this purple, is itself the receptor and the ion channel. And having received this big green ball of the signal, this receptor changes its structural shape and now opens its pore and allows ion channels to pass. So today, the mechanisms that we're going to talk about today are about, well, will reveal that the receptors identified are themselves the ion channels. So I may end up using the word receptor and ion channel sort of interchangeably, because as it turned out that they were the same molecule. I just wanted to point out it didn't have to be so. And when people did the initial experiments, they have to be cognizant of this fact as to whether they have identified the receptor and what is the entity that actually passes the ions. Okay, so let us begin. This is a picture of the skin. This is from a textbook. And the surface of the skin, the epidermis, is dead. Deep within the dermis, there are different structures. Each of these structures are either unsheathed nerve terminals or sheathed nerve terminals, which contain on their tips receptors that would receive different types of signal. And because this is a textbook, here it talks about, cold and heat and gentle touch and pinprick touch and pressure and so on. And all of these have been characterized descriptively as different shapes and different, so some of them lie deep, some of them lie very superficial to the skin and so on. But remember, all of the sensation is done by nerves, and at the very tips of the nerves would be in the membranes, these receptors that would receive the signal. What happens to the signal once the nerve gets it? Well, the entire fiber of the nerve connects to the neuronal cell body that sits here. Okay, this named dorsal root ganglion sits right beside the spinal cord. This is a section of the spinal cord. Dorsal root because this nerve fiber looks like a root coming out of the spinal cord. So here you have the cell bodies of the neurons, their input wires, their dendrites, go all the way to the skin, and on the tips are the receptors. The receptors receive signal, bring the signal to the cell body, and then the output wire, which is the axon, enters the spinal cord. And through a relay system, then eventually enters the brain, where the perception of pain or touch or whatever happens. Okay, so we're going to talk about the neurons whose cell bodies reside here. But indeed, the proteins that they make and the receptors they make and the ion channels they make would all get transported up to the very tips of the dendrites of these neurons. Okay, so now let's introduce David Julius. Here he is as he appeared in graduate school. His history, he was born in Brooklyn, New York, attended public school, undergrad at MIT, graduate school at Berkeley. That's when this photo was taken. In graduate school, he studied peptide hormone processing and secretion in yeast. And then for his postdoc in Columbia with Richard Axel, who was awarded the Nobel Prize later for his discovery of olfactory receptors, that's when David Julius became interested in neuropharmacology and receptor function. What is neuropharmacology? Pharmacology is you use drugs as reagents to stimulate neurons, and you study neuronal sensation, reception, and so on using this reagent. And in fact, Richard Axel identified several serotonin receptors, serotonin being a major neurotransmitter in the brain. He's a faculty member at UCSF currently from '89, and his major focus is on touch and pain. And for his studies, he uses noxious substances from plants and animals, from tarantulas, and as you will see, spiders and scorpions, and this compound capsaicin, which is the chili molecule in chili peppers, and other pungent chemicals in horseradish and wasabi. Here's David as he appeared speaking with some family member right after he got the welcome news. Okay, so chili peppers. This is the structure of capsaicin, and this molecule has excited people for a while. And indeed, in 1912, Wilbur Scoville created a capsaicin calibration scale where he extracted chili pepper extract and alcohol, and in a feat of genuine commitment, diluted it and tested it whether on his own tongue, and came up with this very qualitative scale: bell peppers, jalapenos, habaneros, and then pure capsaicin. Of course, it is not said what the original concentration of the pure capsaicin was. That a 10⁻⁷ dilution works, but you get the idea. And this very qualitative scale is apparently in use even today. Okay, so it's hot. And yet for the receptor, here's what David Julius has to say, that capsaicin had the promise of being a very defining functional landmark. Okay, there was a lot of mystique around this putative receptor before it was found. And there was even an article suggesting that capsaicin didn't really have a specific receptor, that somehow it interacted with the lipid membrane. And every new channel that was cloned in the 90s, there were a lot of channels cloned, people would put this new channel into some cell and ask whether now that cell can be activated by capsaicin. So this is a candidate gene approach. You have a series of candidates, you put the DNA for that gene into some cell that would not normally express that gene, and then you ask, well, now does that cell have some new capability? That didn't work. I should say this lipid membrane hypothesis actually came because capsaicin is soluble in lipid. And a handy tip here, if you eat something that's terribly hot, water will not work at all. Water just swirls it around and activates more receptors and makes you more miserable. But having something with a little bit of fat in it, a little milk, dahi or something, actually does the job because capsaicin dissolves in lipid. Okay, so the candidate gene approach to find the capsaicin receptor was not that successful. So what did David Julius's team actually do? Okay, now the background was that way back in '69, this particular receptor had been found. Transient receptor potential is what it was called, because in flies that were mutant for this particular gene had defective vision. And when people recorded from these, they found transient receptor potentials. So just in terms of what was out there, this existed, and this is going to be a central player soon. But up until 1997, nobody knew of the connection. Okay, so this was the landmark study in '97. Where David Julius's group, led by postdoc Michael Caterina, identified the capsaicin receptor. And they also found it to be a heat-activated ion channel in the pain pathway. And I'm going to now present the data as to how they found this, because this was the seminal work for which David Julius was recognized. So here is the dorsal root ganglion again. And what David Julius's team did is that they isolated the mRNA from dorsal root ganglia, okay, in mice. Now, mRNA is inherently unstable. All cells in the body have an entire copy of the genome. So all the cells have all the genes, but each cell type makes mRNA from only some subsets of genes. And that's how, say, red blood cells will make mRNA for hemoglobin and make protein for hemoglobin, and so on. Now, it was known that dorsal root ganglion neurons are the ones that are sensitive to capsaicin. Remember, they are the ones whose dendrites go all the way to the skin, and the tips would hold the putative receptor. So certainly the dorsal root ganglion neuron cell body must have the messenger RNA for whatever this receptor is going to be. So they isolated pools of this mRNA. Now RNA is unstable, so they convert it to DNA, and you make a complementary DNA library, which is then stable. And then they divided it up in pools. So a cell at any given time expresses thousands of different mRNA transcripts. Okay, it's not that trivial, but they divided it into pools and added these pools to cells that normally were unresponsive for capsaicin. So here are cells in a petri dish. Now this is a cell line. What does a cell line mean? It means it's not the primary neurons taken from the animal. Okay, now it's certainly not a dorsal root ganglion cell line because those are already responsive to capsaicin. But a cell line is taken typically from some kidney cell or some fibroblast and immortalized, so it can be propagated in principle forever. So you don't have to keep going to the primary source. These cell lines are usually more or less homogeneous, so they don't have the heterogeneity that comes from primary cells. They have several advantages, and then you can repeat your experiment in the same cell line again and again. So they offer advantages. And they used a particular cell line from monkey kidney cells that was not sensitive to capsaicin. And the name of the game is, can we now endow sensitivity to capsaicin by introducing, okay, pools of this cDNA library made from the mRNA, which is isolated from dorsal root ganglion neurons. Okay, so they introduced pools one by one, and of course tested by adding capsaicin. And they asked, now do these cells display capsaicin responsivity? What is responsivity? Responsivity is the influx of calcium into these cells. So when ion channels open up in this particular category of receptors, when ion channels open up, calcium from the outside comes in. And since calcium is a very carefully controlled molecule entity inside the cell, influx of extra calcium can be detected. How was it detected? That in itself was an interesting novel technique. So here is the data. Okay, each blue dot is a cell. So these are cells in a petri dish, and the pool number 11 from this dorsal root ganglion without capsaicin doesn't show anything. When capsaicin is added, the cells, a few of them light up. Okay, I'm going to talk about this lighting up in just a minute. This is the calcium signal that's being detected. Now, when this pool gets further separated and isolated, and one single gene gets introduced, this is the VR1 gene, vanilloid receptor 1, because capsaicin is a vanilloid. When vanilloid receptor 1 is introduced by itself, not that much activity, but when capsaicin is added, suddenly every cell is blazing with signal. What is this signal? This signal is a sensor called Fura2. Okay, Fura2 is a calcium sensor. This has transformed biology. It was invented by Chen, who himself was awarded the Nobel Prize together with Shibamura and Calfee for the discovery of green fluorescent protein. So another of Chen's major discoveries was Fura2. So Fura2, when loaded up into cells, doesn't do anything by itself, but when there's an influx of calcium, it fluoresces. So you can immediately see that these cells now have calcium channels opened up. So it seemed then indeed that VR1, this particular gene that was introduced from this library, did indeed encode a molecule that was sensitive to capsaicin. But how heat? We often say chilies are hot. Okay, but what David Julius himself says in one of his interviews is that the idea of heat really just came up through experimentation, where we just kept asking what causes pain. You know, capsaicin causes pain. What else causes pain? What else will activate this receptor? What kinds of things can we throw at these cells that are now expressing the receptor? And eventually we tried heat. And here's what heat does to these cells. Here are the cells just by themselves, warmed up. Just warm medium is flowed over them. Here are the cells with the VR1, but not warmed up. And here is VR1 plus heat, and you can see they light up. So essentially putting in this one gene into this cell line, which previously was neither capsaicin responsive nor heat responsive, now endows the cell line with the ability to be responsive to both. Okay, so this was the seminal finding to have identified this receptor. Now, this vanilloid receptor 1, it turned out was the same as the TRP genes that were found in the fly. Okay, we're same in the same family. Turns out there's a wide family of these genes. And later on, David Julius and Ardem Patapoutian independently found another variant of TRP that is also sensitive to cold, or that is sensitive to cold and menthol. The reason menthol feels cool is that a different TRP gene allows the flow of calcium when either temperature is dropped or menthol is added. So that's how the heat and cold and capsaicin sensors were found. Now, this is the receptor. How does the receptor actually open the ion channel? What is the mechanics of it? In order to study that, David Julius's group took advantage of a very cool finding. Okay, they used peptide toxins from spiders and scorpions and tarantulae, okay, from the venoms, which are known to bind the same receptor. What is the advantage of peptides? Okay, where capsaicin is a chemical molecule, peptides are strings of amino acids, which means they're encoded by DNA, and which means they can be mutated. And selective mutations can be done to ask what part of the peptide is important for what function. And one can really do a very careful dissection of receptor binding, channel opening, and function. So they used these various peptide toxins. And here's a fanciful picture of a tarantula on a chili. Now, what they identified is a bivalent toxin from the earth tiger tarantula. Okay, most of the other toxins are the single clusters of peptide chains knotted by cysteine. They're called cysteine knots. But this one is bivalent. It's two of the similar motifs linked. And this bivalency gave them a lot of power. It turns out that this bivalent toxin is an irreversible TRPV1 activator. Irreversible because it traps the channel in an open state. And here are just some current versus time recordings. Okay, so here is time and here is current. And here, when they apply capsaicin, okay, after a while, the current drops. And thank goodness, because otherwise, imagine if the chilies you ate never ever left your receptors. You would continuously feel the chili heat forever, right? But the chili, the capsaicin dissociates. So that's why the current doesn't continue. This dissociation allows it not to be a very good tool, because you want something to bind and keep the channel open so you can study it. So this other toxin has a slower dissociation curve, but look at this particular earth tiger tarantula toxin. Okay, it essentially never leaves. It traps the channel in an open state, and this allowed studies of structure function. Structural insights came from this landmark study from the same group. It was a landmark not only in understanding the receptor structure, but also in the evolution of Cryo-EM, okay, something called single particle Cryo-EM. And here's what they found. Here is a cartoon from a News and Views written by Henderson, whom I will credit very soon, but broadly in the closed state. Okay, so these capsaicins have 4 transmembrane domains here, and then they have a 5th and 6th domain, a linker loop of which contributes to the pore. And this is a monomer, this is a single molecule, but a tetramer of these forms the cation channel. Okay, so in the open state, when the spider toxin binds, and note how the spider toxin binds in a different place than where the capsaicin binds, but when either of these bind, okay, the pore opens and then ion channels come in. Now, a little more detail, they were able to identify, because they could mutate the toxins very carefully. Okay, they were able to identify that there are 2 gates. There's an upper gate and a lower gate, okay, which open differently depending on how they mutate the toxin. And one portion of the molecule is a selectivity filter, which basically selects what kind of ions go in. So these are the kinds of things that people have to know when they want to study an ion channel. And this is how they discovered that the capsaicin receptor, the TRP1 receptor, is itself an ion channel. Now, Cryo-EM itself was possible because of innovations in the technique. And let me just credit first. So Henderson, who wrote this News and Views, was awarded the Nobel Prize for inventing Cryo-EM with Debouchet and Franck in 2017. And he in his News and Views basically said this was a landmark study because they made many innovations. They used a slightly truncated, more stable form of this TRPV1 construct. They solubilized it better. They used a camera that detects electrons directly, and they could therefore minimize noise. And they used some state-of-the-art software that used statistical methods to estimate particle orientations and arrive at accurate structure. So many innovations came to bear, and I'd like to point out that these innovations did not come from classical biology. Rather, in fact, this Nobel Prize for Cryo-EM, in fact, was a Nobel Prize in chemistry. And you can see various other interdisciplinary approaches that contribute to the availability of approaches with which one can make fantastic insights into biology. And in this case, the structure of the capsaicin receptor. Okay, so I'm now going to switch to the other half of the Nobel Prize, to Ardem Patapoutian. He discovered the receptors for touch and pressure in the skin. So this is the same diagram. We're talking about the same dorsal root ganglion cells, which send out their dendrites into the below the skin, and which send out their output via their axon through the spinal cord into the brain. Okay, now here is Ardem Patapoutian. His background: he was born in Beirut, emigrated to the US at the time of his undergraduate studies in UCLA. He was a graduate student at Caltech. In fact, overlapping with when I was a graduate student there, I've had beer hour conversations with him. And I can say that he looks exactly the same. He looks not one bit different from graduate school. He studied the control of muscle cell fate in Barbara Wohl's lab there. As postdoc at UCSF, he became interested in the development of sensory neurons in touch and pain, and then joined the Scripps Research Institute as a faculty, where he first, in fact, began working with trip channels. And his first major discovery was the cold sensing channel that I referred to, menthol and cold. And he also discovered the wasabi receptor in the same trip family. Okay, and then he focused on touch. And it's this seminal study that I'm going to showcase, Coste et al. Here is a cool picture of Ardem with his son as he is seeing the Nobel announcement on his computer. So this is a really cool picture. Okay, how did they find the touch receptors? In this study, they first identified a cell line that was sensitive to mechanical stimuli. Okay, now this study is almost the complement of the previous study. What did the Julius study do? They took a cell line which was not sensitive to capsaicin, and they sought to endow it with capsaicin sensitivity by putting in pools and pools of mRNA. Here, that approach was difficult. What they identified is they screened through a battery of cell lines to ask which one is naturally mechanically sensitive. And then they decided to find the genes in that. Okay, so completely opposite approach. Now, how do you find whether a cell line is naturally mechanically sensitive? For this, I have to tell you a little bit about the technique. Now, what they did is they applied force to the cell surface via a piezoelectrically driven glass probe and simultaneously recorded from the cell using a patch clamp technique. Okay, these are lots of words, so I'm going to stop share and just show you. Now, here's the cell membrane. Okay, the piezo probe is pressing the cell membrane in steps of 1 micron at a time, okay, without bursting it. So even when I say poke, it actually just means push. Now, simultaneously, a patch clamp, a glass pipette of a very narrow diameter, is touching the surface of the cell with a little bit of suction such that there is a giga-ohm seal between the glass pipette and the cell membrane. So there is no leak current. The only current that can flow through this glass pipette is if in this small patch of membrane that the glass pipette touches, there is maybe a channel. Okay, so they have to keep patching until you find the channel. And when that channel is open, the conduction medium inside the pipette goes through the channel, the charges go through the channel and go into the interior of the cell, or vice versa, depending on which way the current is going. Okay, so the game is that you poke the cell with the piezo and you're patching at the same time and you ask, do you get a current? Okay, so I should say that the patch clamp technique itself was recognized by a Nobel Prize to Neher and Sackman in '91, and it has transformed neuroscience in terms of the ability to record from cells. And here is the data. Now, this is a cartoon. This is the piezo probe that's poking. This is the patch clamp pipette. And here are steps of 1 micron poke. So each trace, x-axis is time, each trace is a push of whatever amount. And here you can see baseline current. And as the push gets deeper and deeper and deeper, you get more and more current measured here in picoamps. So in this cell line, they identified that mechanical responsiveness innately existed. Now, this was in response to pushing. They also applied suction, shown here in mm of mercury. So suction allows them to sense stretch. So there is pressure and there is stretch, which are opposites of each other. And again, steps of stretch also gave responses. And now, completely opposite to what David Julius did. David Julius had a cell line that was not responsive. They endowed responsivity. These people had a cell that is responsive, and they sought to abolish responsivity using RNA interference. Okay? And how did they do this? They examined the transcriptome, which is all the mRNA of these particular cell lines that were responsive. And they manually curated. They chose proteins that were predicted to span the membrane at least twice. And they prioritized either known cation channels or proteins with unknown function. And this unknown function thing is important because somewhere in the literature some years ago, this group, Sato et al. From Japan, had identified a novel membrane protein that was associated with senile plaques in a model of Alzheimer's, which did not have any sequence similarity with anything in the database. And this was sitting in the database. And this was one of the 80 odd proteins that Ardem's group tested. And how did they test it? They used the technique of RNA interference. RNA interference basically uses a complementary RNA. Am I sharing screen?
Okay, yes. Shubha, it's 25 minutes is up. So
okay, or just literally last 2 slides.
Yep.
Am I sharing screen or not?
Yes, yes.
Okay. RNA interference uses a complementary mRNA to abolish an existing mRNA, which would otherwise have become translated into protein. RNA interference itself was recognized with the Nobel Prize. And using the small interfering RNA to one of these candidates that they tested, one of the 80 candidates, they were able to reduce this peak blue trace to this very flat red trace. And thus they cloned what they call the piezo receptor. Piezo, which means pressure in Greek. These piezo receptors are not only present here in the dorsal root ganglia. Each round is a neuron, and you can see the purple signal of the mRNA. These are not only there for touch. There is piezo 1, piezo 2, and a plethora of homologs in the entire kingdom, even plants, because it is pressure and stretch are useful for more than just touch. They are useful, for example, in non-sensory functions, such as the bending of the limbs. You need to know where your limbs are, where your body is in space. Certainly the bladder, certainly the lungs, and for baroreception, which is blood pressure, both piezo 1 and 2 play a role. And red blood cells, they need to sense pressure to adjust their volume. This is a peculiarly interesting function because, okay, the same group, Patapoutian's group, discovered that 1/3 of the African population carries a unique version of piezo 1. And because of this mutation that they carry, their RBCs are more dehydrated than the rest of the population, and they display reduced infectivity to Plasmodium, the malarial parasite. In a mouse model of the same, when a mouse is given the same mutation, Plasmodium infection fails to cause experimental cerebral malaria. So this variant of piezo is actually protective. So you can see how biology sort of allows, advantages where you would not imagine that you would want to have a dehydrated shrunken red blood cells. So that's just a sort of suddenly something bit. And yet, what about structure? It turned out piezo 1 and 2 sequences do not resemble anything that was known. Okay, where many of the proteins that we handle in the lab are maybe 300, 500 amino acids long, sort of broadly transcription factors and so on. These proteins are 2,500 amino acids long. Okay, and here is their hydrophobicity plot. This meant nothing to anybody. There are way too many, some 25 to 30 transmembrane domains. Nobody knew, was this a channel, was this a receptor, what was this? And in order to solve this, Cryo-EM again came to the rescue. This seminal study showed, this is Patapoutian with Andrew Ward, who is a Cryo-EM expert, that there is this protein is a trimer, has 3 propeller-like blades, So this is the Cryo-EM, where the gray is the detergent solubilization effect. But 3 propeller-like blades, okay? And there are at least 26 transmembrane helices, including repeats they call piezo repeats. The cool thing about these blades is they are not flat in the membrane, they are in fact curved. And because they are curved, and because they link to a helical beam and an anchor domain, they are poised to be gated by pressure or stretch. And in fact, many other groups were also working on this. So before this study came out, about 2 years before, this group, Gay et al. From the Tsinghua University in Beijing, also used Cryo-EM to come up with a structure that maybe wasn't as elaborate, but identified 3 propeller-like blades, also curved, and proposed this gating mechanism. This is my last slide. So look at how when the protomers are in the blue position, okay, the propeller blades are curved upward and the gate is closed. And when the membrane is stretched, showing here by the red arrows, the blades flap open and that somehow opens the pore. How this actually happens is not yet known, but certainly this Cryo-EM is a major stride in this, because now we can see this gorgeous structure and examine now by mutation exactly which domain leads to which function. So I will now summarize, or rather close, thanking David Julius and Ardem Patapoutian for their amazing work, and thank you for listening, and to Jyothishman for inviting me to summarize, because it was like a lovely trip through graduate school to read up about ion channels and pressure and how they work and how they communicate. All of these references will be in the chat box. I encourage students to read them. And remember, biology is not just for biologists. Everybody contributes.
Okay, thank you very much, Professor Shubha Tole. I would sort of ask Ankona to put her things together. There could be one question. Anybody from the Zoom or the audience here? Yes, there is a question there. So maybe I'll just walk over.
Hello. Yeah, it is a nice talk. So, so given that we know the mechanism of touch or pain receptors, is it possible to make artificial touch and pain receptors that will help in medical science also?
A good point. What will help in medical science is to inhibit pain, or inhibit the excessive pain from pressure. So there are conditions where even a light touch is very painful. Okay, so understanding the way the receptors work at the domain level will allow designing specific drugs that don't have extra side effects, but specifically block the processing of pain in response to a certain type of touch, or unnecessary triggering of pain where the signal should not have triggered it. So we don't need new receptors. We need new tiny drugs that block exactly what you want to block. And we already have receptors, right? So does that help?
Okay, yeah. So thank you, Shubha, for that answer. For all other questions for Shubha, please type it on the chat box in Zoom or YouTube, wherever you are. And we would be, or she would be happy to answer those. And
I'll be happy to answer. Sorry, I went a little over.
That's